I thank you. I thank you for the invitation, and to me, it's again the second opportunity within a year to be able to speak to Brazil. Once I was in Brazil during November last year, in a city called Barretos, and here it's doing it by satellite or by video, and I'm very pleased to be able to have this opportunity. And I understand that my audience is made of scientists, right? If I understand well. Yeah. Oh. Okay, so I'll be talking today about the drug discovery in general, the basic principle of drug discovery, and also I will be talking about the activity, the drug discovery activity here in Canada, talking about many different centers that started or that initiated programs in drug discovery. So first of all, let's talk a little bit about the traditional way to produce a cancer drug or to find new drugs. Now we have three main ways traditionally, three main paths by which we will discover or isolate drugs. The first one is isolation of active principles from plants. The second one is total synthesis of natural products. And the third one is, of course, to structurally modify drugs that we know that are already active. Next slide, please. Next slide, please. Now, breaking with this tradition, right now we have major advantages in terms of drug discovery, and we can say that we are tremendously aided by, first of all, the ability to generate small molecule libraries. By small molecule libraries, we mean synthesis of many different types of molecules. And importantly, the significant increase in the number of newly identified gene sequences. And finally, the use of structural methods for the detailed characterization of ligand-protein interaction site have significantly enhanced our ability to design and produce a new drug. And next slide, please. We also have new methodologies, new models that allow us to discover drug at a faster pace. Next slide, please. There are two main aspects, two main phrases that we have to define when we talk about drug design. And from the time of those of you who can remember the story of Alexander Fleming, who discovered penicillin. From the time of penicillin, where we accidentally, or there's a very rare word that it is serendipitous. We used to serendipitously discover a new treatment. For example, at the time of Fleming, it was the discovery of an area by the microbes that were by the fungi that would that led to the death of the bacteria that was surrounding the area. And then this has led to the discovery of penicillin. And it was what we call a serendipitous discovery. So now we work more with a rational drug design approach, and this can be defined as two different strategies. One of them is what we call the ligand-based design. The ligand-based design is based on the fact that we have knowledge of the bioactivity or the activity of one particular molecule. Then we can rationally make changes into this molecule. What we call ligand is a molecule. In other words, we call it ligand because it is a molecule that can bind to the target. Now, the second approach is a structure-based design, which is a strategy based on the 3D structure of the targeted protein or the target itself, and this can be obtained by X-ray or by solution NMR. Now, these strategies, although the definitions seem to be very simple, are very complicated, and I would say time-consuming. And we have unraveled the complication of these strategies now by using molecular modeling, or what we call computational chemistry. So modern drug design is more rational, and that's what we call rational drug design. Now, next slide, please. Now, the paths toward discovery, most particularly, this is most particularly related to the development of cancer drug, is divided into many stages, and I will go quickly through the stages. In stage 1, it is the case where we can select an agent from whatever the mechanism, from high-throughput screening, and then we determine the antitumor activity of this particular agent. In stage 2B, we study the mechanism of action of this agent, and in stage 3, we study the toxicity, the tolerance of this particular agent in the dog. And before, many years ago, it used to be the monkey, but the second species can be the rat, the dog and the rat. We most likely now do, most often do now, perform our studies now in dog and rat, in terms of what we call GLP toxicology. Stage 3 is required in order for us to move on to stage 4, which is the phase 1 to move into human. Before this phase, I will show later, you have to apply for what we call an IND, and then later on move on to stage 5, which is phase 2. And phase 2 is the time where we define whether this particular molecule is active in human. And then ultimately we have stage 6, which is phase 3 clinical trial, and following phase 3, approval from the FDA in the United States, for example, can be applied for in order to market the drug. So next slide, please. This is a process that is, next slide, please. That is rather lengthy, and I always shock the students when I tell them that to develop a drug, it takes an average of 12 to 15 years. And in terms of funding required to develop such drug, it is about $1 billion from the discovery stage to the marketplace. Now, as you can see from this diagram here, the step discovery, the step that we call discovery, is the step at which we design the drug, or we screen to identify the drug, and then we synthesize or modify or do what we call lead optimization, which is taking the compound that shows activity and try to optimize it, to perfect it, in order for it to bind optimally to its target and induce its biological activity. Now, after this stage, we move on to what we call preclinical development, which is in vivo model and ultimately GLP toxicology in mice. And then this will be followed by an application for investigational new drug. And following approval of this investigational new drug application, we can move to phase 1. And ultimately, as I mentioned before, phase 1, phase 2, phase 3, and ultimately the drug will be approved. And this process is lengthy. It takes around 12 to 15 years. Now, I go back here to say that the first stage, how we fund the activities, usually, most particularly now, we'll be talking about Canada. In academia, we are funded by what we call grants. The granting agencies are agencies that fund projects without expecting any financial return from the project. They are charitable organizations, not-for-profit organizations. This pool of funding allows us in academia to be able to design drugs, to develop new methods, to test the drug on new models and so forth. And ultimately, for the drug to advance from the preclinical development to the phase 1, usually investment from a private partner is always required. Now the preclinical phase is all the way to what we call GLP toxicology, the one I explained before, which is done in the dog and the rat. We're talking about an average of $300,000 for one drug to be able to be tested, and for us to be able to test one drug and ultimately move on to phase 1 clinical trial. So next slide, please. Now, in order for us in cancer, I'm specifically here referring to cancer drug, one parameter that we use often for examining the effect of cancer drug is the IC50. IC50 is the inhibition concentration for 50% survival. So in cancer, we usually take, as we do in my laboratory here, is that we grow tumor cells on a plate, and then we administer the drug candidate. And if the drug candidate is potent, it will kill the cells or prevent the growth of the cells. So the concentration that inhibits 50% of the growth of the population of cells, we call it IC50. So IC50 is for us an index or a parameter, a value that allows us to compare whether the drug that is modified or is the structure that is obtained from the modification of a lead drug, whether it is better than its ancestor or worse than its ancestor. So we use these IC50 numbers to say whether a drug is potent or non-potent. Okay. Now in vivo, we have, next slide please. Now in vivo, we have now models that are what we say clinically relevant. One of them is what we call the Harrow fiber assay, which is developed by at the NCI, the National Cancer Institute of the United States. And it consists of taking human tumor cells and putting it in a fiber and then implant this fiber into an animal that is immunodeficient. But I would like to insist more on the right picture of the animal. This is an animal that is immunocompromised. As you can see on its back, you can see a tumor burden. And this tumor is composed of human cells. So this is one of the most clinically relevant model because we are able to grow human tumor cell in this animal. Recently, we have begun to use a model called PDX, which means patient-derived xenografts. That means that the tumors derive directly from the patient, and then they are implanted directly to the animal. So patient-derived xenograft is one of the most clinically relevant model to test new anti-cancer drug. Next slide, please. Now, when we were referring to the model, the mechanism of funding of drug discovery at the early stage, I was referring to foundations, to organizations that fund research. In Canada, we have the Canadian Cancer Society, and the Canadian Cancer Society has a particular unit which we call the Canadian Cancer Society Research Institute that funds research on a competitive base, and investigators with a project will submit this project to the what we call CCSRI, the Canadian Cancer Society Research Institute, and the project will be evaluated by the panel and rated, and then there will be a certain percentage of winners based upon their ranking in the panel. So I took this particular slide to show you that in 2016, in a total of $28.1 million invested in basic environmental constitutional aspect of cancer project, 14.9 million of this total funding was devoted to diagnosis and treatment. So there's a large portion of the basic biochemical and translational funding that goes to new drug and diagnosis, mechanism of action of new drug and diagnosis. So in Canada, we pay important attention, as you can see from this pie graph. 75% goes on to treatment from these particular organizations. However, we have other organizations, for example, the Canadian Institute of Health Research is one of the organizations, we call it CIHR. It's one of the organizations that also fund cancer research as well. We have the Prostate Cancer Canada, we have Leukemia and Lymphoma Canada, and also we have the Cancer Research Society that funds research, that support research at the discovery stage. And of course, companies, projects also can be funded by companies, by private companies at the level of discovery. So next slide, please. Now, in Canada in particular, over the past decade or more, we have got an emergence of new institutes, new entities, new units that are devoted to translational research. Translational research means research that is devoted to convert basic science into useful drugs or useful treatment for patients. So an example here is in Montreal, an institution called the IRIC. It stands for Institut de Recherche en Immunologie et en Cancérologie. It's a research institute in immunology and cancerology. And IRIC is one such model which is a not-for-profit institute, where research very recently has led to the discovery, a unique discovery of a molecule that is capable of stimulating the growth of stem cells. This is needed, as you know, in terms of organ generation, the context of organ generation, regeneration, and also, of course, in the context of leukemias, where we would like to stimulate stem cells. So next slide, please. One compound recently identified by the University of Montreal is called UM171. It's an agonist of human hematopoietic cell, and it was discovered about 4 or 5 years ago. And this particular molecule now, as you can see in the bottom of the slide here, is now being advanced to clinical trial. So it's an example of our ability to discover and advance drugs from the bench to the clinic in institutions that are not for profit, that are academic institutions. The IRIC is an institute that belongs to the Université de Montréal, University of Montreal. So as you can see, this beautiful structure here, UM171, has now been advanced to phase 1 clinical trial. Next slide, please. Another example of institution, not-for-profit institution, is the Alberta DNA Repair Consortium. It's a group of investigators. This has been initiated by one of my colleagues, Dr. Michael Wenfield, and the group assembled a number of researchers. It assembles numbers of researchers who have vested interest in DNA repair. But however, with one purpose, is a translational purpose, in order to be able to identify mechanisms in DNA repair that can lead to the cure or treatment, of cancer. Now, the group has been created 2 years ago, and they already have a pipeline which is being developed for the treatment of colorectal cancer. So the Alberta DNA Repair Consortium is another example of a group or an organization within a not-for-profit context that is capable of working on designing new molecules for the treatment of cancer. Next slide, please. Another important institution that has been created over the past, I think, 7 to 8 years was the CDRD. The CDRD stands for Center for Drug Research and Development. So the model is an excellent one. It's a business model whereby, I say business, but please keep in mind that it's a not-for-profit organization. Their mission is to source, evaluate, develop, and commercialize both small molecule and biologics. And this particular group of researchers are located in Vancouver, in the cancer agency, in an academic institution. And however, they are an independent unit, and their activity is twofold. Number one, they evaluate molecules. If you have a project, you have a problem, a particular problem that you would like to resolve, or you would like to translate into a drug, they will analyze the problem, and they will propose a solution to you, and all the way from the design to the commercialization. Importantly and interestingly, from this organization were born several spin-off companies. One of them called Sukara Therapeutics, one of them called Sitka Biopharma, and the Sitka Biopharma has created or designed a nanoparticle for drug transport in cancer. And there's also Keros Therapeutics. All of them were born from the CDRD, and as well as Zymeworks. Zymeworks is a company that produced antibody drug conjugate, which is classified now as what we call biologics. So this is we've been talking about Quebec, Montreal, and Vancouver, and then the consortium for DNA repair is in Alberta. It's in a city called Edmonton. Now let's see what we have in Toronto. Next slide, please. So in Toronto we have the MARS, and the MARS is another organization that is based on the similar model, with the same mission to source, evaluate, develop, and commercial both small molecules and biologics. And the MARS has been very successful in creating new spin-off companies, new small pharmaceutical companies. They produced a new product called Celax, which is a nanoparticle, and also from the MARS were born about many startup companies, and most particularly they produced projects. They worked on projects that allowed to hire more than 4,000 people since its existence. There's also another company that was born from the mouse, which is called Consta Pharmaceuticals, and they are developing alpha-fetoprotein as a carrier for anti-cancer agents. So as you can see, the activity of drug discovery and development has evolved very strongly from not-for-profit organizations that involve academic people, and you can see that from academia we can also translate to the private sector as well. Now, next slide, please. There is a strong training component in the activities of these particular organizations that we are referring to. Activities can be training students at the PhD level, at the master's level, and also, of course, as postdoctorate. Postdoctorate, the model from the CDRD in Vancouver, the model is mostly oriented towards postdoctoral fellows. So they recruit postdoctoral fellows who are able to participate into the different projects, several projects that they are working on. So in our case, next slide please, we have here developed a very specific training called the McGill CIHR training in drug development. And this is organization where I am still the director of this organization, which is called DDDP. And the program seeks to train students on the principles and aspects of drug discovery and development. So next slide, please. In the history, the structure of this program is such that we have an executive office, we have an academic training committee, we have a scholarship committee, we have a workshop and development committee, and then we also have a membership committee. Next slide, please. All these units work together to deliver the program. The scheme that you're seeing here is that the program is made of what we call members. We have DDTP members, and these DDTP members are students who are successful in winning fellowship from the program. And then we also have the professors who enter the program through selection by a membership committee. So the activity, next slide, please. Next slide, please. So the activity of this program are such that students receive a fellowship from the program. However, they have to specifically work on drug discovery related subject. Next slide, please. So the academic component of the program is made of different symposia, classes invitation, and most particularly we favor student mobility. We fund the student to travel, to visit different centers, but each of these trips or projects or trips that they have to take, these trips, they must be towards conferences that are strictly relevant to drug discovery and development. Okay, so next slide, please. Now, the program is interdisciplinary. It encompasses pharmacology, chemistry, medicine, and those are the three main disciplines that we bring together, or we still keeping together, in order to be able to talk about drug discovery. As in fact, we need the chemistry, we need the pharmacology, and also ultimately translate into clinical trial, which is where medicine has a very important intervention here. So next slide, please. We have now, with the program, evolved into organizing, as you could see in yellow in the bottom, every year an international drug discovery and development forum. And as an intercity at the last forum, we have Brazilian investigators present in the forum. We organize this forum every year, and it concerns anyone who is involved, any unit, companies, etc., private and academia, who are involved in drug discovery and development. And this year, the main theme of the conference was challenges in personalized medicine, in using bioinformatics, informatics to be able to predict the response to cancer drug. Next slide, please. So now, speaking of students, the new challenges for the students is the diversification of the field and needs for skills for drug discovery and development. So students must gain experience now in management, in regulatory, in marketing, business development, manufacturing, and clinical trials, IND submission, etc., etc., etc. Those are the new fields that in which there is a need. Those are what we call the soft skills. Those are the skills that students now need in order to be able to effectively contribute to the challenges of the pharmaceutical industry. So all these now are being resolved by the different organizations that have been talking to you about from Canada. Each of the organizations, such as CDRD, for example, they have a training component where they give an opportunity to the students to learn and acquire these particular skills. So next slide, please. Next slide, please. Recently, we have here developed or incepted what we call the CTB drug discovery platform. And our hospitals, which were located for 100 years, 170 years on the campus, which was not far from where I am located now, merged and moved to this place, this new place here, which we call here the Glenside and the Super Hospital. And within this hospital here, we created this what we call the CTB drug discovery platform, and it's a complement to what we had initially, that what we called the drug discovery or drug development training program. This particular platform here, what we call the drug discovery platform now, includes a chemistry laboratory, an NMR laboratory. We have the most sophisticated instrument in terms of NMR analysis, and also multi-imaging as well. Next slide, please. Next slide, please. Here's the group that initiated the platform. And you can see me in the middle here. And our purpose in this platform is to assist all investigators from the institute in designing new drug, and also, of course, the development as well. And we advise, we consult and advise on translational research in our institution. So the drug discovery platform is 2 years old, but however, it was born from the Cancer Drug Research Laboratory, in which we have been devoted our time to design new drug. So I'm going to briefly talk to you a little bit about the type of drug program that we have here ongoing. Next slide, please. Next slide, please. Within the platform, I must highlight that we are able to analyze samples from human tissues or from animal tissues using NMR with what we call the HR mass technology. Within this technology, we were not able before to be able to define or to detect or to profile responses to tumor using intact tissue without disintegrating the tissue. So we have now technologies to be able to analyze in situ the drug itself or any other metabolites that can be in that particular tissue. Next slide, please. This is complemented by another instrument that we call the MALDI mass spec imaging. As you can see here, this particular instrument can image, it can create image of compounds or structures or chemical, not by the photons, but by the mass. As you can see here, if you point your MALDI to the mass of 746, this is the image that you have on the left. If you're looking for the mass of 904, you can see the image right after on the right. That gives us the possibility to analyze in situ without disintegrating the tissue, the presence of molecules that we would like to analyze, for example, like lipids, peptides, proteins, and more importantly, the drugs themselves. So instead of doing in the past radio labeling of the drug so that we are able to see this particular drug in the tissue, then we have to disintegrate the tissue in order to be able to image it. Now, using the mass spec MALDI technology, by the mass only, we are able to track, image the distribution of this particular drug. Okay, next slide, please. In our platform, we have been able over the past to study and design new drugs. One lead drug that we have is called ZR2002. And ZR2002 is a dual EGFR and DNA targeting molecule. It's a molecule that can hit the EGFR and it can also hit the DNA as well. So it's a single molecule with the ability to reach 2 different targets, thereby sensitizing the cells to tumor. So let me give you a very quick outlook of the molecular basis of this. Please next slide, please. Next slide, please. Now, this type of molecules, they are designed to target a marker called EGFR, the epidermal cofactor receptor. The epidermal cofactor receptor is a transmembrane receptor which is overexpressed in many tumors. Please, next slide, please. In many tumor types, next slide, please, including the lung, the breast, the colon, the bladder, and the prostate, etc., etc. And what we did is that we designed compounds to block these particular receptor, which is overexpressed in these diseases. And instead of having a single drug capable of blocking only EGFR, we designed the molecule to contain another function that can damage DNA, thereby enhances the potency of this particular drug. Next slide, please. The molecule is based on a structure that is known to be active against EGFR, which is called the quinazoline. The clinical drug now that are targeting EGFR are the Iressa, which is also called gefitinib, and also Tarceva. As you will see, next slide, please. As you will see, these particular molecules, they do not contain functions that can damage DNA. Here, as you can see here, is an example of functions that can damage of DNA adducts that can kill the cells. And this is produced by a molecule called temozolomide. So our plan was to fuse this temozolomide with the quinazoline in order to obtain a compound that is dual targeted. Next slide, please. So we successfully, here you can see how the molecule, this molecule can bind to EGFR, to the EGFR pocket. As you will see, if you look at the red part of the molecule, it is exposed to the mouth of the receptor, the binding site of the receptor. So therefore, we are able to make modifications in that part of the molecule without affecting the potency of the molecule. Next slide, please. So in the next slide, what we did is that, in fact, we designed a molecule, and if you can click one click, give one click, one click, one click. Just click once, or no, just one click. So that go back, please. Go back, please. Go back, please. Okay, just go back. No, please, please. Next slide. Next slide. Next slide. Just hit the arrow on the keyboard. Just hit the arrow. Well, it's not working. Okay. So what we see here is that this particular molecule is designed. Please, could you put it back, please? Please. This back, please. Okay. So this particular molecule is designed to behave as a quinazoline inhibitor of EGFR and to release this red moiety, as you see here, that is called methyl diazonium. This is directed as targeting DNA. So as you can see here, the molecule is a prodrug, what we call a prodrug, for an inhibitor of EGFR, and also can generate the methyl diazonium species, which is a DNA alkylating agent. Next slide, please. So we are able to design a complex molecule. They remain very small, as you can see, and they are complex enough to degrade into multiple inhibitors or to hydrolyze into multiple inhibitors, and ultimately a DNA damaging agent. So next slide, please. So our idea here is, unfortunately, you can animate it, is that we would like to revolutionize combination therapy. As you can see on the left, in the basic mechanism or the basic formula for combination therapy is that you have a drug 1 that is directed at target 1, and then you have a drug 2 that is directed at target 2, and then you have a drug 3 that is directed at target 3. What we would like to do and what we are developing now is to have a single molecule called combi molecule that can hit all the 3 targets at the same time. So this is the type of drugs that we are investigating and we are developing here in our platform in Canada. Next slide, please. So we are almost at the last one. So in conclusion, to make an overall view, a broad view of the activities of drug discovery activities in Canada, in many different provinces of Canada, we can conclude that the academic milieu, the academic medium, is very active in drug discovery. Here, as you can see, I take the example of 3 cities, Montreal, Toronto, Edmonton, and actually 4 cities, Edmonton and Vancouver. And you can see that the interaction between the private sector and academia is very strong and synergistic and productive. And we have to mention that many of these not-profit institutions or organizations or networks that are created from these networks well-born spin-off companies, in other words, from the academic sector, can derive private companies that create jobs and positions for our students. Importantly, the infrastructure and tools for drug discovery here in Canada are being modernized. Here, where I now, is an example of the modernization of our infrastructure. Here, in this building here, we have equipment in the amount of $100 million in total, and this equipment are new pieces of equipment that allow us to accelerate drug discovery. Now, very importantly, and more importantly, these developments, these activities are not separated or uncoupled with activities of training. They are in parallel, they are hand to hand with training of students onto first regular classical programs, which are master's, PhD, and postdoctoral fellowship as well, but also into very special programs like the one I have described to you, which I created with my colleagues, my colleague Dr. Papadopoulos here 9 years ago. So we have special training programs and we have the regular curriculum. So this has concluded this short survey, this short view on drug discovery activity here in Canada. And I have given you an example of discovery of breakthrough that was made in each of these individual cities. And also I introduced to you our brand new platform that now is equipped with sophisticated instruments capable of accelerating drug discovery. So thank you. Thank you very much. And it was a pleasure for me to be sharing with you the drug discovery activity here in Montreal.
Yes, and very, very useful here in Brazil.